So welcome everyone. I wanted to welcome you to this session on building enhanced fair workflows through the use of PIDs within, so that's persistent identifiers within and between interoperable research tools. And just wanted to start just by saying, welcome to the Research Data Alliance International Data Week. SciDataCon. I wish we could be there in person, but most of us, or all of us, I believe, are virtual right now. But welcome, thank you for joining us. We have a lineup of speakers really to speak about interoperability across the research lifecycle through research workflows, and it's something that's near and dear to something that I look at from different angles. I come from the American Geophysical Union. I'm Christopher Erdman, and we see at least some of the sort of challenges related to that when we're sort of publishing papers. So when we're working with authors, we can see from our end some of the advantages that I think many of our speakers will actually talk about of how they're trying to address this, that we can benefit as well from. And so just from my own sort of personal experience working with, so I'm the data steward for the American Geophysical Union, and I think so. One of the things that Xiaoli actually said in one of her slides is papers are not enough, and I think that's where we're sort of all on. I think where we're all headed on the same page is that we believe that there's much more, to sort of the research, sharing research, so sharing all the sort of outputs related to that. And I think this is what this session is all about. Is so we have someone that needs to be unmuted. So yeah, just sharing all the different research outputs that are out there, and really unpacking, sharing all the different aspects, including data and software. We'll hear about samples as well, and just sharing multiple sort of different outputs from the research process. And I think that's what we're trying to tap into on our end. And I think at AGU, I think we can benefit from this, but we also see that working upstream, working with authors so that they sort of understand these things. I think there's still a lot to be done as well, working with authors so they can understand how they can share these outputs. And so I'm really looking forward to hearing from all these authors, all of our speakers, about how they're sort of addressing different aspects, the challenges involved in interoperability, and really linking all these things together so that we have sort of more efficient workflows, and where we can sort of tap into all these advantages of shared metadata. I think I just also wanted to thank Rory McNeil, who really brought all of us together. He brought, I think, all of us from different angles, which I think is really impressive. So thanks, Rory, for doing that. But I think we'll start off with, we switched up the schedule. If you're looking at the schedule right now, we'll start off with some really some of these domain related examples, and we'll start off with Jens. Actually, if Jens, if you want to introduce yourself and then present as well from your perspective.
Sure. Thanks, Chris, for the introduction. I'm Jens Klum. I'm with the Commonwealth Scientific and Industrial Research Organization, where I lead the Exploration Through Cover team group, and I'm also the vice president of the International Generic Sample Number Organization, which is something that will feature in a moment in my presentation. And I think I'll dive straight into it. Just bear with my slight brain fogginess today because of COVID, but I'll make it through this call. So I wanted to give you a brief presentation about the International Generic Sample Number as a persistent identifier for physical objects, and convince you that it's not just a new label on a box. So the elevator pitch is that we all know how difficult it is to track samples across institutional and system boundaries and unambiguously link them with data and literature. So IGSN provides a persistent identifier system for physical samples through a globally unique resolvable identifier that is compatible with other persistent identifier systems. IGSN persistent identifiers are already used by major research centers, universities, and government geological surveys. And as an identifier, it is endorsed by scientific publishers. And IGSN persistent identifiers are now available through DataCite to all DataCite members. It's one service and one subscription for all these different flavors of persistent identifiers. But the angle that I'm coming from is that samples are at the heart of research, not only in the geosciences, but in many other of the natural sciences. They come in many shapes and forms, but they are often our anchors into reality. The data that we derive come from samples, and it's important that we know what we're talking about. And these samples are mobile, then they don't sit in collections until the end of time. They move between collections, they move between labs, they move between institutions and organizations. So to be able to track them through a globally unique identifier is a really important feature. So as I said in my elevator pitch, IGSN is a persistent, globally unique, web resolvable identifier for physical samples and specimens. There's some debate on what is a sample, what's a specimen. Not for now. It's domain agnostic, and the samples can be any material from anywhere. Why did we want this? This was initially an idea developed by Kerstin Lehnert from Lamont Doherty Earth Observatory of Columbia University when she started compiling geochemical data from the literature. And there are some precious samples that have been analyzed many times in different labs with different methods. The example on the left-hand side shows you one sample with many names, but you can't be sure it really is the one. And on the right-hand side, the map shows you locations of samples named M1. So that is one name, many samples. Clearly, M1 is not a good name that allows for unambiguous tracking of the sample through collections, data, and literature. So this is where the idea came from, that we needed something that is globally unique and should also offer more than just global uniqueness, but also be actionable. So this is where IGSN came from. And just in a nutshell, here's an example from my home organization, CSIRO, from our spectral reference library, where we have this piece of kaolinite. It has an identifier, so we know which exact piece we're talking about. On this piece, we measured reflectance spectra. Those reflectance spectra are now housed in our data access portal and identified through a digital object identifier. And some of these spectra appear in the literature and publications, which in turn are by themselves identified by DOIs as persistent identifiers. And the underlying infrastructure of the identifier issuing organizations like DataCite and Crossref allow us to cross-link between all these objects. So we can, if we find something in the literature, we can go back to the data and we can go back to the specimen to find out what we need to find out. Like in this case, was this really kaolinite, or did they mix this up with something else? But we also use it to track samples. So we had a very successful geochemical field campaign in the Nullarbor Desert of South Australia, where we used IGSNs pre-printed on labels and encoded in QR codes. And then we used an app from the framework called FAMES to build field apps, field data collection apps. And we could use the app to scan in the code of the pre-printed label at the moment when we were taking the sample. And by using this app, we could pre-populate a lot of the metadata in that collection process because we knew who we were, where we were, what time, what mission, and so on, which type of sample. This was tailored to the specific expedition. And so this really sped up the process of documenting and this samples enormously. We had the streamlined process improved the efficiency of sample handling by 100%. You can see the helicopter in the background. We had to use a helicopter to access the sampling locations because they were very far away from any roads. And so if you have to go to that length, time is money. And this kind of streamlined efficiency really pays off in many respects, both in the resources, but also in this case, also in the quality and completeness of the data. Some of you might have heard about IGSN before as an independent organization. IGSN still exists as an organization, but when it comes to the technical infrastructure, IGSN and DataCite have entered a partnership to have IGSNs minted through DataCite. And we entered this partnership at the end of last year, and we are in the process. We're now around number 5 in the transition process, where we are starting to transition our agents onto the DataCite infrastructure. And the next step will be aliasing the existing IGSN handles into the DOI system. But we are also starting to welcome new users on board. And as I mentioned earlier, any DataCite member organization can now start minting IGSNs through DataCite infrastructure. For the community, this means that the role of the IGSN organization changes. It becomes more of a host for what we call the community of communities. The idea of this community of communities is to have different research communities build their own protocols and best practices around sample description and identification, and acknowledge that the community has varying levels of skills with respect to persistent identifiers and metadata. Having this organization will enable more cohesion around the use of IGSN and thus enable greater research discovery, innovation, and the advancement in the use of samples. So if you want to hear more about that, you're welcome to contact Rory Edmonds at DataCite, who's the DataCite Samples Community Manager, or any of the people below. And on the right-hand side, a few more links to follow. And thank you very much for your attention. I'll hand back to Chris.
Thanks, Jens. And I think you already have a question. I think we're going to have 20 minutes of discussion later, but we can use the chat for some questions. You already have one, but I'd like to move on to Anita, who's, I think you joined us, Anita, right? And oh, there you are.
Yeah, I'm here. Hello.
And if you can introduce yourself, thanks, Anita.
Okay, yes. Hi. So my name is Dr. Anita Bandrovsky. I am a part of the Department of Neuroscience, and specialist in the Department of Neuroscience at UCSD. I am also, as a conflict of interest disclosure, I am a founder and a CEO of PsyCrunch Incorporated. And I am currently at the Berlin Institute of Health doing a summer sabbatical. It's funded by the Charité. So I think that is relatively full disclosure. My information is here. So talking about these persistent identifiers, sorry, I need to move you guys out of the way. These are research resource identifiers, and I just wanted to bring up 2 slides just to show you why we're relevant and why we're important. So this is a standard paper in biology. You know, you get your title, you get all of this stuff. In the methods section, you might get descriptions such as this, you know, mouse anti-flag from Sigma. And this is a reagent that these authors are describing. And yet when you go to Sigma, first thing you'll notice is that the name of the company is not actually Sigma, it's now Millipore Sigma, and that changes actually quite frequently. There are multiple matches to this particular descriptor. And at this company, which means that this reference to a reagent is actually not unique. And so this paper, if someone wanted to reproduce this paper 1-for-1, they would not be able to do so. And for those of you who are not biologists, antibodies are one of the classes of reagents. You probably have heard a lot about them in terms of you know, their role in combating COVID and other things. They're incredibly powerful reagents. They're also things that bodies make. And so they, you know, can be generated by many different people, animals, and these are very powerful, but they are incredibly variable. So I think you saw probably early in the pandemic where some of the tests for COVID were actually inaccurate because the antibodies were failing. So this is just one of the classes of reagents. RRIDs actually are not an entity type, not in the same way as IGSN. It is a group of different repositories and registries. One of the registries, which is an authority of a particular RRID type. So one of the registries is called the Antibody Registry, and it is the authority for antibodies. And this particular database provides the identifier for each of the you know, commercial and non-commercial antibodies that are available. Out of that list, you know, there are about 2.5 million that we've cataloged so far that are available to the community. So just to kind of give you a point of number for scale. And so anyone in the community can buy one of these 2.5 million things. And then identifying, you know, one of 2.5 million things by name is obviously quite problematic. So the identifier from the antibody registry, which is the authority, is shown here. It also has, you know, the entire RRID syntax actually includes both the company name, the catalog number, and that identifier from the authority. And that is the case for many other authorities. So for example, the rat genome database is the authority and the registry, in fact, for rats, while Cellosaurus here is an authority for cell lines. AdGene is the authority. And actually it is the repository of plasmids, which are little snippets of DNA, mostly circular. But all of these things are things that you might be able to buy, bring into the lab and use. Now the question is, how can we get authors to actually use these RRIDs in their papers? What the first thing we did back in 2014 is we put everything into a single website from all of these different authorities and sources. And so the author can be directed to that website. They can type in their search, and then they can be confronted, for example, with the fact that there are 17 antibodies for that in that previous example. So if the author is actually using RIDs, they can very quickly confront this problem that you saw. So when we first published, and this is just on the first 100 papers that we found with RIDs, and then as a control, we looked at the same journal, but the issue before we started asking for RIDs, you could see that for antibodies, we had about 50% of the cases, like you saw in previous slides, where we could not find the antibody, whereas with RIDs, a little bit unsurprisingly, but in any case, it brings the percentage of identifiability of a single product up to about 90%. And that was true for the other resource types as well. And then something a little bit more surprising came when there are issues that are reported with a particular research resource, like in this case, a problematic cell line. There was a contamination event that was noted where actually one cell line called HeLa, and this is the first cell line that was ever derived. Great movie by Oprah. Thanks to Oprah, actually. If anybody is interested, The Immortal Life of Henrietta Lacks actually talks about the genesis of this particular cell line. But this cell line actually had contaminated at the source another cell line, and some of those contaminated samples went out. So these are biological entities. They divide, they grow, they do all kinds of interesting things. And so, these are things that can certainly become contaminated. They can be problematic in other ways also. So we actually looked at, one of my students, Juliana Babic, actually saw that about 16% of papers out of 150,000 papers that had used cell lines had actually used a cell line with one of these warnings, not all quite as bad as this one. But when authors were using the RID, which, in which case they would confront the warning, that percentage, dropped by 66%. So we thought this is really cool, because we're actually not just identifying what's in the paper, but we're also cleaning up the literature of, the uses of some of these bad cell lines as an example. So we can actually use the system of RIDs to actually push the author to cleaning up what they're doing. Okay, so I just wanted to show you what some of these records look like, and I think one of the other speakers actually makes use of some of these records in I think probably JSON format. There's a couple of different formats that you can use. This N2T net, this is one of the resolver systems that is available, that RIDs are available through. This is the name to thing resolver from the California Digital Library. Another one is identifiers.org. I didn't specify that here. And then there is our native resolver, which I think most of the publishers use. But this is one of the tools. So there's a tool set of things that set of entities that authors use. And so this is a flow cytometry system. It's just a big instrument coming from BD. And what you can see is that in this summary report, you have a set of kind of persistent and appropriate metadata for this particular entity. And the other thing that I think you're hopefully seeing here is that there is a set of mentions in the literature. So these are citations or mentions where we've been able to pull out what the authors are actually saying. So here in this 2021 paper, they are saying that these authors from in this particular journal are saying, the facts caliber BD Sciences, and then here is the ID. And so we know that this is, some of these 379 things are actually text mined, but the verified ones using RID can also be accessed alone. So there are quite a few authors that have actually done this already for at least this type of tool. And the one thing that we always tell people, especially in the space of, in the authority space, is that, you know, RIDs are just numbers. You know, we have some infrastructure around these numbers, but they're not magical numbers. They do not bring magical compliance from authors. What they will do is they will simply identify the entity. And so a lot of the you know, the places that support, like, for example, Thermo Fisher Scientific, this is one of the antibodies that is, again, identified with the RID. This is a little pop-up box to tell you what that is. This is on all of the antibody pages at Thermo Fisher. Here is a very common mouse repository. And now this is, you know, our newest actually compliant repository, where the RID citation with the little, you know, icon just explaining what the initiative is, you know, it is available right there on the third row of all of this different rich metadata about this particular mouse. And this is kind of a famous mouse, so lots of people use it. Okay, I think I'm probably going very slow. Let me move on. The publishers, the way that they ask for these things is either through instructions to authors, letters to authors, or through checklists. And this is one of the kind of more recent efforts out of Science magazine, where, you know, there is a particular supplemental file that is available and required for all papers in Science, called the Materials Design Analysis Reporting Checklist for Authors, which authors are, you know, made to fill out with each accepted publication. And, you know, lo and behold, the first two items on that checklist are antibodies and cell materials, where, you know, RIDs are being asked for specifically. And so people do get asked for these things. And we actually just crossed a milestone about a couple of weeks ago. There are now 500,000 RIDs that are available in the literature from about 45,000, over 45,000 journal articles. So this is what the this kind of looks like. And, you know, this is definitely dominated by a few of the large journals like Cell Reports, which is the big kind of +1-like journal from the Cell Press family. Things like eLife and Journal of Neuroscience is kind of our biggest society publisher, and many others here. This is what they look like in, again, in a particular journal article, where there is an author who says, hey, I used this particular mouse. You can see here's the name of the mouse. Here's another name for the mouse. And here's the identifier, which makes it unambiguous. And that information based on this identifier can actually be called from a backend service called the resolver service that I pointed you to before. And that information is available for curators to verify that this is the right mouse. And when that's verified, this name of the mouse again is associated then with a particular publication. And of course, we are not quiet about what we do, so we actually tweet about that information. So I think I'm going to wrap it up here, and I'd like to thank everybody. And I think the questions are until the end, right?
Yeah, there's a discussion 20 minutes. Thanks, Anita, for your presentation. But there is a question in the chat if you wanted to answer Matthias's question there. So yeah, thanks, Anita. We're moving on to our next section, which is about workflows and interoperability. And our next speaker is Shali, if you're ready to present. Thanks. And if you can introduce yourself.
Hello, everybody. Yes, thank you for having me on this session, and happy to talk about the project we're working on. Right. So apologize in advance if I sound a little bit zombie. I've been awake since 4 a.m. I'm in the Netherlands right now. So yeah. So I keep it short, and we'll briefly introduce the Implementing Fair Workflows project, going through the motivations and our approach to address some of the problems arise in fair compliance throughout the research process. And I will introduce some of this a series of integrations among services and platforms that we will be building inside of the project that is aimed to boost persistent identifier adoption and to optimize metadata usage, thus laying the groundwork for a fair workflow. Just general information, this is a 3-year project funded by the Thompson World Charity Foundation, led by DataCite and the Max Planck Institute for Empirical Aesthetics. The DataCite will be obviously focusing on the metadata part of the workflow, and the research team at the MPI will be carrying out the research project, which the fair workflows will be built on. They are the example. So this will be a neuroscience project, as suggested by the subtitle of the of this project. Is a proof of concept study in the field of consciousness. So there is also a range of project partner organizations, which I'll go into detail later in the presentation when I talk about integrations. So starting from the motivations, first and foremost, Chris have already picked this one out. That is like paper is not enough, even though frustratingly, it is still very much the predominant research output that is recognized in the field of consciousness research. And we all know that you only tell a little part of the research story, and it's very hard to comprehensively evaluate research study based on papers alone. And secondly, like many other fields, consciousness research is growing in terms of the scale of collaboration, and the experimental protocols and data that emerge from these studies are becoming increasingly more complex, which aggravate the reproducibility problems. And meanwhile, the existing technology and support structure is not reaching the researchers. It's not that they don't exist. There are tools and infrastructure available to support open and fair research, but the adoption is very fragmented. So researchers may know to submit preprint or start a pre-registration, but very rarely we don't know any case where fair compliance is considered and applied throughout a entire research lifecycle. And finally, for the sake of practicality, in order to motivate open practice adoption and fair compliance, researchers need something that's tangible, that's concrete examples of this workflows putting into action, and that are actually easy to implement. So although this project is based on neuroscience research, we hope the experience and outcome will provide value to scientists out there in other fields that are facing similar challenges. So in one sentence, the general idea of the project is to implement an exemplar fair open workflow based on the reality of the entire research lifecycle. And to achieve this, we'll have to take care of both the practice side of things and the metadata side of things. And we're approaching this project from these 4 aspects, starting from the workflow development. Here we're talking about the research workflow and the metadata workflow that's behind the scene, which will support the fairness of the output as the research proceed. We do this by mapping all identifiable entities. This is including the investigators, the involving institutions, the project itself, and the potential outputs that's planned to be produced during the research, and the relationships at the beginning phase of the project. And in another word, to start fairness by inception, at the inception stage. And then we work with the project partners to implement a series of system integrations where they are needed and that cater to the needs of the research workflow that we develop to capture these information, the metadata of the entities and their relationship to, with each other. So this will produce a wealth of metadata, and we will feed all of this into the PID graph for reuse. I'll talk about PID graph in the next slide. We will be using this the PID graph to build a dashboard to demonstrate the availability of these outputs and their connections. And on top of that, there will be a separate effort to communicate the project and push for the adoption of the outputs, including best practices recommendations. So very briefly about the PID graph, it's a data model that represents research through nodes and relationships, where each node represents an identifiable entity, and the relationships are provided by trusted sources. Here are the PID providers. And so with that in mind, we can take a step back and look at the workflow. So as I said, the project was set on the existing PID metadata infrastructure that's already been being used by the research community. This diagram visualizes 3 layers between the PIDs to the end user interface, the dashboard. So layer 1 are some examples of the PID that would be expecting, including investigators with ORCID IDs, organizations with ROR IDs, projects with RAID IDs provided by the ARDC, and outputs with the DataCite or Crossref DOIs. And the layer 2 is a combination of aligning all the metadata behind the identifiers and mapping them onto the PID graph. So in other words, making them queryable through the DataCite GraphQL API, basically. And the layer 3 is that the user interface that we're talking about. So this interface will also, on top of showing the availability and relationship of entities and outputs, you will also track the usage and citation information of the outputs. And so all this will be achieved through coordination between project partners, obviously, to foster interoperability across systems. And here we'll enter the meat of this integration. So this is the potential workflow. This will provide a general framework for planning and evaluating the existing support and the gaps in the infrastructure. So in the project, we have a number of partner organizations providing a great range of different research services and platforms together. They basically support all of the research activities throughout the life cycle of the project. So we have identifier systems on board, ORCID, ROR, Crossref, and DataCite that provide the basic metadata interoperability. And we have Dmp Tool that facilitates the authoring of the data management plan. CDAR is a metadata workbench that enables researchers to build domain-specific metadata templates. It also provides interoperable components for templates to be used in different stage of the research. And the open science framework covers the registration and the preprint workflow. Dryad and Cronus Hub covering the data deposition and manuscript submission processes respectively by the end of the research process. So these are some of the existing integrations, so meaning there are already metadata flowing through these systems. And in this project, we'll be building a series of new integrations to facilitate association between research outputs. For example, so association between a data management plan and domain-specific metadata template through the integration between Dmp Tool and CDAR, or so like CDAR and FIAD integration will allow this template to associate with a specific data set. And of course, CDAR will also integrate with the data site to enable DOI registration for the metadata products hosted on CDAR platform to provide the basis for domain metadata reuse. So we'll also be enhancing some of the existing integrations between DataCite and Dryad and DataCite and Dmp Tool to improve the metadata that's being exchanged there to make them more richer and more accurate. On top of that, we'll also introduce project level identifier RAID and work with ARDC team to make it interoperable with other types of identifiers. So on DataCite side, we'll be taking the opportunity to review and supplement the current schema documentation to review and evaluate particularly properties within the related identifier property element within this property. So for example, we'll be considering adding pre-registration and metadata template resource types to the control list, and also provide more guidance on assigning relationship types when using the related identifier inside of that dataset metadata. And we'll also be establishing interoperability with RAID to integrate RAID into the PID graph, and working on strategizing some approach for bridging generic metadata like the ones DataCite method schema facilitates, and the domain-specific metadata, which are arguably more meaningful for researchers. So to summarize, the goals for the integrations I was just talking about, first and foremost is to support DOI registration, to systematically capture all types of outputs throughout the research lifecycle and generate rich metadata, and to enhance metadata completeness, particularly through this related identifier property, which provide contextual information for better discoverability and better like assist to reuse. And we want to use this opportunity to promote adoption of metadata best practice to provide better guidance for integrators when assigning resource type metadata and relation type properties to improve the quality of this metadata and support more specific aggregation use cases. And last but not least, we want to provide as many as possible examples, lessons learned, and roadmaps for future integrators to easily become part of the solution for fair research workflow. So just one sentence back at the dashboard. The dashboard is planned to be a variation of the DataCite Commons. Currently, the DataCite Commons support aggregation on the level of work, researcher organization, and repository. For the project, we will be designing a new interface that aggregates information on the project level. So some immediate next steps on all three fronts. DataCite will be starting to map resource type and relation type to the current workflow for the project. For the research team, they will be building the domain-specific metadata template. And lastly, the project partners is kicking off the implementation work packages starting this month. So yeah, that's the project. Happy to have shared it with this group. And this is 3 years project. We are 3/4 into the first year. So we'll be keep connecting with the community and to keep iterating on our approach and seeking feedback from everyone who's interested. So yeah, check us out on all of these channels, and thank you for listening, and happy to answer any questions.
Yeah, thanks.
Later.
Yeah, there'll be questions in the chat. I did want to say, Shelly, your sound was off and on a bit during the talk. So I don't know if you want to turn off your video, if that trick will work with your sound, but it was a little off sometimes. So maybe later for the discussion.
Okay, thank you. So, wasn't aware of that.
We could still hear you, but it was just a little bit off. So thanks, Shelly, and then Maria. I think you're next.
All righty. Oops. All right. So just let me know if the sound goes wonky for me, and I will turn my video off. So someone will have to unmute and tell me. So my name is Maria Pratzelis. I'm with the California Digital Library. We're part of the University of California. I work in the UC3 department, where I work on initiatives around research data management. I run the Dmp tool, so I'm going to talk about the ways in which we are using the Dmp tool combined with PIDs to improve interoperability and to exchange information between systems. So I'm going to start off, assuming most people are familiar with DMPs. I'll do it really quick, and then we'll talk about how we PIDified the DMP, what PIDs we've added to the DMP to add structure, and then some specific case studies where we're actually utilizing the information in a DMP and exchanging it between systems. So for those who aren't familiar with the Dmp tool, we're a free open source community supported tool. We've been around for 10 years. We're a platform for the creation of DMPs. So it provides guidance, best practices, and templates for all of the big funders in the US, also international funders as well. So we are widely used, and increasingly we're international in scope. We have every week I get new emails from folks around the world asking to join the tool. I always like to give a shout out to our editorial board. These folks are the ones that help me keep the tool up to date. We are a staff of one product manager, me, one developer. So I really rely on the community to keep things updated, to give me feedback on features, keep us in line. So I owe a lot to these dedicated data librarians. So I'm going to switch and talk more about network DMPs. This is really the meat of what we've been working on at the Dmp tool for several years now. We've really started from the beginning, coming up with the structure of how we were going to express data management plans in a structured format, coming up with the common standard with the RDA working group, and now moving that into the Dmp tool. And now we've got that infrastructure in place, and now we do the fun part of taking that on the road, integrating with other systems, and continuing to build on it. So really the goals of a networked DMP are to share all that good information that's in a DMP across all the stakeholders, to link metadata, share it with repositories and institutions, can allow for notification and verification,
automated compliance. And really the big picture goal, if we do everything right and we build these systems carefully and thoughtfully, the big goal is to lessen the administrative burden on researchers and grant administrators, because this stuff should be done behind the scenes with information exchange so that people don't have to duplicate entry and so forth. So for those who aren't familiar with DMPs, even across funders, even across nations, they usually have the same types of information contained within them, which has really rich information about research projects. So that includes things like what type of data is being collected, what are the tools, software, and code used to manipulate or analyze that data, what are the standards and data documentation used. Most important part for most funders, issues around preservation, access, and data sharing. So any requirements around data sharing, where are you going to share it, when are you going to share it, what license are you using, etc. And finally, roles and responsibilities. So who's in charge of this, contact information, who's the data curator, what are the sort of roles in which you're going to actually follow through on the promises of a data management plan. So those are the kind of common elements across the board. All that rich information is in a DMP. Our job was to structure it so that we could share it, update it over time. So what we did, we partnered with DataCite, and last year we released they released the Dmp Id, which is a unique identifier. It's a DOI for the data management plan. Within the Dmp tool, these are just a few of the PIDs that we've added. It could include things like IGSNs. It could include RRIDs. So anything associated with the project, really. So that includes ORCIDs for researchers, the RORs for all the affiliations, associated outputs, the data repositories that are going to be the eventual recipients of the data, the funder information, all of those behind them are identifiers. So we really believe that the first step was to put a PID on the actual data management plan, and then you would have the ability to create unbreakable links to the associated outputs as a project expands over time. So this GIF is supposed to demonstrate the purple dot in the middle is the DMP. All the individuals on the left, that's the team. As you can see, it grows over time. Behind that is all the rich ORCID metadata. And then the part I really wanted to demonstrate is the data sets and the publications. So you can, this is supposed to represent associated data sets that were eventually published as part of this project and associated publications. So you can see through the use of identifiers, we're able to track that, and it evolves and changes over time. So now I just want to talk about three specific ways in which we're using networked DMPs currently. We are also partnering on the Fair Workflows project that Xiaoli just talked about, which is a really awesome project. I'm super excited about that work. I'm not going to talk about it because she just did it, but we are involved in that, and it really touches on a lot of the same things about connecting outputs, creating best practices so that other implementations down the road have kind of this manual for how to do it. So the first one I wanted to talk about is an integration that we just built with Rory from RSpace. I'm going to talk about it very briefly because Rory's going to give you more information and a demo. But just briefly, this integration, again, uses the exchange of identifiers in order to connect the planning stage of a project with the active research stage of a project that happens in an electronic lab notebook with the eventual data deposit. So in the case of this integration, we partnered with Harvard, so they're sending it to Dataverse. That could be sent to Figshare, it could be sent to Dryad, Zenodo, whatever repository the user is pointing to. So basically we're exchanging related identifiers and PIDs to make this happen and to facilitate the exchange of information. And Rory will show you a video so you'll see more specifically about what that looks like in a second. The other project I wanted to talk about is the Fair Island project. This is an NSF eager grant. We are just wrapping up the first year of this project. It's similar to the FAIR workflows, and since I'm heavily involved in both projects, it's a really great way to make sure that we're really aligning our best practices and working together and sharing resources. DataCite is also a partner on the Fair Island project. So what Fair Island is doing is we're kind of two main kind of avenues of work. The first is developing optimal data policies for facilitating open science practices and open data practices at working field stations. And the second avenue is looking at the technical infrastructure necessary to support an environment where all data that's collected on a field station is made openly available as quickly as possible. So those are kind of the two areas of focus of this project. And I'm just going to talk about the technical infrastructure piece, which has been my area of focus. So the use case that we are looking at, so whereas Fair Workflows is looking at one specific team in cognitive science, we are looking at field station directors. So there are kind of our main use case. So in sort of our user story that we developed for this, we've identified the problem through interviews with other field station directors. And basically the sum was that field station directors have limited insight into the research outcomes and subsequent impact of all of the good research that's done on their field station or reserve. So basically researchers come in, they do their time at the station, collect their data, what happens to that data, where it goes, is it shared? Are there publications that come after it? Directors have little insight and ability to track that. Some of them have developed kind of ad hoc ways, but nothing is systematized and really formalized so that they could do long-term tracking. So the directors that we spoke with were really saying that they need to demonstrate the scientific impact of their station so that they can demonstrate the value to their administrators, to their board, to their funders. We all have to answer to someone, so they really need data and metrics to prove the impact that their station is having on science. So some of the questions that we're trying to answer are: what research outputs resulted from time spent at the stations? Where was it deposited? Were they cited and reused in subsequent publications? Where on the station was the data gathered? And was the research that they supported in compliance with university or government policies? So just briefly, to some of the infrastructure that we built to support the facilitation of this information and exchange between systems really involved integrating with existing systems and utilizing the existing DOI infrastructure. So in the Fair Island workflow, we worked with a reservation management system that's run by the University of California. So basically what happens is, as a researcher wants access to a field station, they fill out a form requesting a reservation. That information is sent to the Dmp tool, pre-populates a DMP. Then the researcher comes in, finishes their DMP, gets a Dmp Id, and with that marker, we can then link it to the eventual outputs that are associated with their project and their time at the field station. So switching to kind of the third area, and this is something that we are still kind of in the early stages of thinking through and building, and it really ties to a shift in the way federal funders are handling data management plans and data sharing specifically. So for those who aren't in the US, this may not seem like big news, but for US researchers, the new NIH data management and sharing policy is a very big deal. NIH has said that researchers now are required to come up with a plan, but more importantly, that they need to maximize the appropriate sharing of scientific data. So data sharing needs to be the norm, not the exception. So that's a big shift for these folks. They haven't had to operate in that way before. So a lot of folks in this community are thinking about ways in which to help researchers comply with these policies and to help ways for institutions to make sure that they're in compliance with these policies. So we're trying to think about the ways in which we can use identifiers and the exchange of information from data management plans, from grant IDs, in order to create a new system that facilitates tracking of information and the connecting of a plan to research outputs. So that's really kind of what we are thinking about, what is next for the Networked Dmp. So we're really responding to these increasing funder mandates for data sharing, for stakeholders like the field station director, who really needs to know what research is currently happening on their station, what outputs are created from that time. So we're building on baseline work from things like the Fair Workflows project, from things like Fair Island, from things like our integration with RSpace to build a new service that has a tracking for research for improved planning, compliance, and really metrics for impact. So that is what I've got, and I will answer questions at the end, and I'm always open if folks want to chat more about this, shoot me an email, and yeah, thank you.
Thanks, Maria. And you do have a question from, I think, Anita in the chat. So, but in the meantime, Rory, if you wanted to share your slides and present and introduce yourself.
Okay, thank you, Chris. Okay, can everyone see my screen?
We can see it. Yeah, there's
Okay,
a bar,
great.
There's a bar down below if you wanted to move it. I think it's that toolbar, but
Yeah,
otherwise we
okay.
can see it.
Okay, thanks. So yes, so thank you. And first, thank you, Chris. It's what Chris said at the start is not entirely accurate because he also helped put this together. So thank you, Chris, for your support. And it also, as you'll see as I go through the presentation, it's been great to cooperate with many of the people that are here today. And in particular, I'd like to give a shout out to Anita and Jens, both of whom have given really valuable advice on the project of helping to PIDify what we're doing. So before I talk about
Rory,
yeah,
move the bar up into your slide. If you move it down, because now it's blocking text. So, and if you can move it farther down, but it was blocking text.
Yeah, do you know how to move it farther down? Because
well, it's not blocking text now, so let's continue on. Maybe.
All right, fine. Okay, great. So the problem I think that we're addressing here at RSpace, and I'll talk about RSpace in a second, but really it's the problem that everyone's talking about today. It's the comprehensive capture of both experimental and sample data, and I think both. It's important to differentiate experimental and sample data, but then also bring them together. So it's the comprehensive capture of both experimental and sample data, and the recording, sharing, discovery, and reuse of the data and appropriate metadata. And this is hindered by limitations of research tools, and siloing of data sets that the tools produce. So the objective, which again I think is really the objective of that we're all talking to in one form or another, is to enable workflows supporting the experimental reproducibility and comprehensive capture of sample metadata. So a picture tells 1,000 words, and I'm the CEO and co-founder of RSpace, which started out life as an electronic lab notebook, which is, for those who don't know, and I suspect probably everyone does, but it's an increasingly popular category of tools which were originally designed to help people who work in labs transition from paper lab notebooks to more digital forms of record keeping. over the years, RSpace has transitioned to become more of really a much broader function of a research data management engine. And the way we've done that is not by trying to build everything into RSpace, but just the opposite, which is the theme of today, which is trying to become interoperable with other kinds of tools and resources that people use. So now we really focus more on the ecosystem, very reminiscent of the kind of things that Xiaoli was talking about with workflows which enable passage of data and metadata, including sample data between tools. But I'm going to talk about that at the beginning. Let's focus on the experimental data. And RSpace is, if you think of it from a researcher's point of view, inside RSpace you can document your experiments, but it's very important to be able to associate with the write-up of the experiments all of the data that might have been used in connection with the experiment. This could be big data like sequencing data or imaging data, which is probably kept on an institutional store somewhere. It will definitely include small data, documents, spreadsheets, possibly PowerPoints, things like that, which might be kept on resources such as Google Drive or Dropbox. We also support association of specific types of data, like but through integrations with animal colony systems to be able to associate your animal data. We've just done a nice integration with an equipment management and scheduling app. So you can associate the equipment that was used to produce your experiment. You can associate that with your write-up as well. And also, we are planning, we're working on, I should say, an integration with FAMES, which is the field data acquisition app, which Jens described in his earlier presentation. So you can associate lots of kinds of data with the data that you use to write up and the data sets that you produce inside RSpace. We've got some other neat integrations I'm not really going to talk about today with things like Slack and Teams with Protocols.io. Actually, I'm going to talk about that, so you can link to and import protocols from Protocols.io. We've got this is just a bit off topic, but we've got a fun new integration with Jove, the Journal of Visualized Experiments, so you can actually play, link to and play Jove videos inside RSpace, but that's not really today's topic. And then, so that's kind of from the researcher's point of view, but research data management is also important. And so we started out in order to facilitate reproducibility. One of the first things we did was integrating. You can see, you can get the whole point about RSpace is it's interoperable, so it's very easy to get data out. So we started out by integrating with repositories like Dataverse and Figshare. We're now working on one actively with Dryad, and Zenodo is coming next, so that you can deposit your data in these repositories for query and reuse and reproducibility. And then more recently, as Maria said, we did a nice integration with Dmp Tool, which is now Dmps have become the second go-to tool for research data management. So through this integration and the other integrations we have, RSpace can become a value-adding conduit in the research cycle from preparing from the preparation phase, and RSpace takes center role during the active research phase, and then passing on data sets for archiving, storage, and reuse. So that is our story generally. Now let me see if I can play the video that Maria mentioned, just as an example of the kind of thing that we've done, the integrations with Dmp Tool and Dataverse. To do that, I'm going to stop sharing. Share my screen again. And just a sec, sorry. Apologies for that. Need to do one more thing. All right. hopefully you'll be able to hear this as well. Here we go.
View of the Dmp tool and RSpace integration, which facilitates linking together the data plan and project outputs with minimal effort. I have here my list of DMPs in Dmp Tool, and I'm recording my research and storing the data in RSpace. I can easily browse DMPs inside of RSpace. I can filter by my DMPs and public DMPs, and can select which DMP to import. Importing into RSpace will store the DMP as a PDF inside of the document gallery, which I can easily view, as well as reference and link to within RSpace. The DMP will also update in RSpace when someone on my team makes changes to it in Dmp Tool. Now, here's my research data for this project. I am ready to export my work. This is a straightforward process within RSpace, as I can directly export to an external repository such as Dataverse. I'll make an HTML export and provide the information Dataverse requires. After the deposit is confirmed, I can view all of the research data in Dataverse. Most importantly, a direct link to the exported materials on Dataverse is then passed back to the original DMP in Dmp Tool as a unique and permanent related identifier, which supports traceability and fair data. This identifier is also recorded on the landing page for the DMP. This landing page serves as a public-facing page listing all outputs related to the specific project. As additional datasets are published, these citations will be automatically added to the landing page, keeping the research materials and DMP in sync throughout the research process.
Okay, I'll just go back to my presentation and okay, so that was an example of tool interoperability, in this case between 3 tools, and you saw how the the DOI was automatically assigned on deposit into Dataverse back to the DMP, which is a nice workflow, and it happens, as Maria said, behind the scenes, so it makes it convenient for researchers. So what we're working on at the moment is something I haven't mentioned up until now, which is we have a very nice new mobile-friendly, highly visual sample management system, which has been deeply integrated with the ELN. And so many of the things that we're doing now are around this new inventory system. And one aspect of that, one critical aspect of it, is today's topic, which is incorporation of PIDs. So this is kind of a slimmed down but also updated version of the earlier graphic that you saw between the preparatory, active research, and archiving phases of the research, and you see the data management plans here. And so if you think of of external data, and what we're going to do now is have more general PIDs, late data site DOIs, associated with experimental data. The Dmp Tool video was an example of that. And then we want to support 2 workflows, and I think this is critical because this keeps coming up. So better association of general PIDs with experimental data, and then deposit into general repositories. So that's one workflow. The second workflow is equally important, and I think Anita asked a question about this in the hopefully we can chat about this later. But domain, it's also important, in particular in the context of samples. It's critical, as Anita was demonstrating with her RIDs, that domain PIDs get into domain repositories. Not generally, they aren't so relevant in general repositories. So the workflow that we're working on at the moment is to support association of domain PIDs like RIDs and IGSNs with sample data in our sample management system, and then subsequent deposit into domain repositories. And something else I'm not really going to talk about today, but also in addition to another aspect of this, in addition to domain PIDs, is incorporating metadata schemas from other databases and allowing filling out of those schemas inside the inventory system and then deposit. So this slide is a bit off topic, but I wanted to give you a sense of what a brief sense, at least to show you that it's real, of what the inventory system, the sample management system in RSpace looks like. So on the right here, you see an example of a sample template which has been populated, and then this would actually appear at the bottom of this, and this is a domain schema which was taken from Chebi. So again, the workflow is to incorporate domain schemas inside RSpace, to populate them, and then to export them to the relevant repository. And as part of that flow, to also incorporate relevant PIDs in the record within RSpace inventory. And I would definitely like to say a huge thank you to both Anita and Jens, because incorporating information about different types of PIDs requires a different interface, and it requires different workflows. So they've both been kindly providing advice on how we can enable incorporation of in the inventory interface of information about different kinds of PIDs and how that fits into workflows. And then finally, because really kind of jumping back between PIDs and interoperability, I'd like to just give you a quick sense of the, again, FAMES is the field acquisition information management system, which Jens showed that they were using with his group, and then associating an IGSN in FAMES. So one of the workflows that we'll be supporting is to ingest PIDs from other tools into RSpace before they're then passed on, say, to a domain repository. So at the moment, we're working on an integration with FAMES, and in order to do that, we have matching templates for data capture in both FAMES and RSpace. And then the workflow this is what Jens described. An interoperable notebook is used in the field to record sample metadata at the time samples are taken, including assignment of an IGSN offline, key point, and on multiple devices. So this is what Jens showed. Then on returning to a network, data synchronizes across all devices, and using the APIs, the metadata, including the IGSNs, will be passed from FAMES through and ingested into RSpace. And then once the data is in RSpace, you can do all the stuff you do in RSpace. You can associate it with other kinds of data that you used in your experiment. You can write up the experiment, etc. Then step 2 would be, to export back to FAMES for further work, if there's further work in the field, and then the cycle repeats. Then I guess unfortunately, this thing, here we go, I'll get rid of it. And then, as you saw, because of the pre-existing RSpace integration with general repositories, and also now domain-specific repositories, it's possible to deposit both the general and the domain-specific metadata in those repositories. So again, a different workflow from the one that I showed with Dmp Tool, but really achieving the same objectives. So RSpace is very much in the same spirit as the Fair Workflows project that Xiaoli talked about, and really, in particular, thinking about the FAMES workflow, which will enable enhanced capture of field data and workflows via offline capture of data, enhanced findability by association of metadata and PIDs with the data, and then advanced availability and reusability by streamlined deposit of more and higher quality data in both general and domain-specific repositories. So that's me. Thank you very much.
Thanks, Rory. And I don't know if we'll be cut off at 11, or it's 11:30 my time here. But Yeah, 30 minutes past, but I'll try to get our next speaker in, who unfortunately couldn't be here. It's Rowan from CurveNote, and I will share a video from Rowan. So, oh, sorry, let me share my screen and actually check these boxes so you can hear the sound.
Hello, I'm the founder and CEO of CurveNote, and today I'm going to be talking to you about improving metadata and reproducibility during the research process. And I'm going to be talking about this in the context of the tools that we are building at CurveNote. And those are authoring tools for scientists and research teams that integrate directly with computational notebooks like Jupyter Notebooks to improve and accelerate scientific communication. And by accelerating science communication, I'm mostly focused on how we as scientists work and how we share that work. For example, in traditional publishing workflow, sharing happens months after the scientific work is actually completed. And this is traditionally a PDF behind a paywall. And this, of course, has changed massively over the last several years as we move towards open access workflow and sharing preprints much earlier than journal articles. And so that acceleration of sharing and communication is starting to move upwards towards the scientific process, and as we work with open preprints. And really, the trend and a lot of the topics at this conference are about how to formally share and cite and collaborate on more and more of the scientific work that's going on. So the problem design and study design, data collection, analysis and interpretation, and that's much of the work that was traditionally sort of behind the scenes of a traditional publication. And so, for example, data and sample and software citations, pre-registration of studies, micro-publications to share components of work, and new ways to share preprints in progress. So that communication is moving upwards in the scientific process. And there is, of course, also a lot of informal sharing in this space as well. And as we move upstream, that communication does become much more sort of informal, in quotes, with lab group seminars, blogs, and tutorials, Jupyter Notebooks or computational notebooks in general, software documentation, open educational resources. And this is an area that we've been working a lot on at CurveNote, is really how to bridge the gap between this informal communication as scientists are working on their work, on their science, and how to bridge that to more traditional scientific artifacts. And our focus is especially in the context of Jupyter Notebooks, which are used in data analysis and interpretation, and thinking through the balance between archiving, interactivity, and reproducibility. And what we've seen is that helping researchers bridge this gap not only saves them time, but can have a whole lot of other benefits around metadata collection and reproducibility. So today I'm going to give a bit of a few examples of some of the tactics that we've been taking at CurveNote to help improve this informal scientific communication as researchers are working, and including helping them sort of capture rich scholarly metadata, integrating their computation and communication tools, and maintaining that metadata when they actually share their work, and allowing them to share their work in new, open, interactive, and in progress ways. And so I have a couple quick screenshots to show that of what we are doing at CurveNote to capture this metadata, as well as sort of the persistent IDs behind the scenes, like ORCIDs and DOIs and RIDs, and integrate computational workflows from Jupyter Notebooks, as well as how you share that work, so that we can improve reproducibility, as well as have the chance to share sort of more interactive data. And the goal of this is to make it easy enough so that you can share this throughout the research process without a huge amount of overhead. And so I'm going to show a couple screenshots of working in CurveNote. And so this is CurveNote. It's primarily a writing tool for scientific content with integrated references and citations, as well as collaboration through comments and version control. And here you can see sort of someone writing and creating some cross-references to figures and equations. And that information is stored as data so that it is accessible when you actually share your work. And in this case, sharing it as a website. And so this is more of a browsing experience in this sense, where folks can click on sort of a cross-reference to a figure and bring up that rich information both about the figure and any other cross-references inside of it. And that sort of really improves the reading and browsing experience. And this also works with more traditional citations through DOIs. And so those are directly built into CurveNote, allowing you to search through your entire reference collection without using your mouse. You just type cite and start searching through your collection. And then we can add rich information like abstracts pulling from DOI services like Crossref or OpenAlex. And so that rich information is again shown and sort of incorporated from these scholarly sources like DOIs, ORCIDs, RORs, OpenAlex, RIDs. And I know Anita will be talking much more, or has already talked about RIDs for biological research. So I'll let her take that one. But I think the real piece that I think is beneficial is showing that immediate benefit to adding that rich scholarly metadata while you're actually writing. And this encourages authors to actually add that information as well as validate that it is correct by able to sort of click on it and quickly see. The other major piece that we're focused on is going through every piece of content in CurveNote and making sure it has a unique ID. And this opens up all sorts of workflows to be able to integrate outputs from Jupyter notebooks that can be interactive visualizations directly in an article. And because they have that unique ID, comments and versions can sort of be passed around wherever that output is embedded. And that really aids in the collaboration and iteration process. And we've architected CurveNote to separate the content as data from the presentation of that content. And this means you can very easily change a template when exporting to a specific journal, although when exporting that loses any sort of interactivity and a lot of the rich metadata. And so some of our authors are choosing to share their work as websites that require minimal setup to sort of self-publish that work. And these allow authors to share more intermediate work and sort of more of the behind the scenes information. And of course, because it's online and not in a PDF or a Word document, there are a lot more options to explore with embedding interactivity and data directly into the presentation of results. And so this is an example of running a Jupyter notebook and showing portions of the results in narrative content, which is, of course, very helpful for educational use cases and tutorials. And so these are a few of the strategies that we've been taking at CurveNote to improve scientific communication. By integrating metadata and persistent IDs while writing, we're focusing on the immediate as well as the lasting benefits of additional data, like those abstracts and the descriptions and tags that are coming from the community. And we're sort of pulling for that reproducibility in science by integrating computational workflows directly into how we're sharing work. And ideally, by making this easy, as easy as possible, we are improving the iterations and collaborations when sharing that scientific work. And so I'm really excited by how folks are using CurveNote today to share more of their science, share it sooner, and share it more openly. So thank you so much for listening to my presentation. And please do reach out. I would love to hear from you. All right, thanks so much. Bye-bye.
All right, thanks, thanks, Rowan. So I'm not sure. Oh, I had some black boxes too, I guess. Thank you, Matthias. But I don't know if we actually have time because our session was we're set to the end of it right now. So I don't know if someone from RDA is going to come on and kick us up, but maybe we have additional time for 5 or 10 minutes for questions if anyone has any questions or comments. Did you see any, Rory, when I was playing the video? I wasn't watching.
I didn't see any. I think there were a couple that have already been answered. I didn't see any new ones, but then maybe I missed.
Okay, the session doesn't start for another 30 minutes. But yeah, I think we'll still want to give people breaks. So, but if we didn't get any questions, I think just Matthias did. If we didn't get any, Matthias wanted to speak just a little briefly to RAID. If we wanted to give him a chance to talk about that. So Matthias, you have the floor.
Great, thanks, Chris. Well, I mean, I already wrote most of my words in the chat window. So yeah, RAID, the research activity identifier, has been around for a couple of years, but there's still a lot more work that we need to do on it to make it really useful to our partners. And so we are very interested in talking to people who would be interested in using RAID to work out what they need in terms of a project identifier, or how they would integrate it into their systems and workflows. So the Fair Workflows project is a great example of that, but I have seen other projects here that might be interested in RAID as well. So please reach out to me. Interestingly, Rory, you know Sean Ross from Fames?
Yes, I know Sean very well.
Yes, Sean has just joined the ARDC as the RAID product manager.
Yeah,
he's still working on.
I heard about that. That's great. Yeah, congratulations. Yeah.
Yep. So he's still working on Fames though, so don't worry. So yeah, I think I will leave it at that. Thanks for the time, Chris.
Yeah, no problem. And I guess like part of our abstract, or the session description too, was sort of some of the next steps. And I just wanted to conclude and say that in some ways, listening to all the projects, it comes back to, I think, something that I've said often in other talks, is it takes a village to make interoperability work. And so it seems like from some of all the other projects that we've seen, is that it takes really, all these groups coming together to make it work. So it's like considerable investment of your time, but also, the funding, we also need to sort of tap into funding, and just seeing that across the projects, and seeing how much work goes into interoperability, just working together. But I guess I also wanted to leave with this too, is that I see from my end, from researchers, that we just demonstrated a number of solutions, and maybe I'm not describing this well, but the sort of overwhelmed or perplexed researcher that is having a hard time keeping track of all these sort of projects out there that they can take advantage of, and sort of that sort of outreach and training aspect. From my end, I often see researchers say, I'm fair, I'm fair with my research, and then we're seeing this also in a survey that we're doing on open science in RDA through the Shark group. But when you delve down further, researchers still don't necessarily know what fair means, and they still don't necessarily know what the landscape is like. And so I guess at the end, I would just leave that with everyone, as saying, how are you sort of engaging with the researchers, and how you sort of getting to them in various places, and identifying your particular solution, so that they can take advantage of it. Because I know that that's challenging. I find from my end that researchers are still unaware of some of the things that they can take advantage of. And I guess if we have a little bit of time, we could pass that question around. But I don't know if, so Jens, you have a hand up.
Yeah, so my experience with introducing the geochemists to the Fames app a few years ago was that they liked it because it made their lives easier. So if a new thing solves researchers' problems, then they're happy to adopt it. If it's additional work, then it has to be very, very well motivated to and then people will find a way around. Or it has to be invisible. If it's invisible and it just works, it's also fine. But it has to be one of the two. It either solves a problem or it's invisible, and otherwise you're facing an uphill battle.
Thanks, Jens. Yeah, I think that I mean, that really captures it. But you know, I again, I think it's just like trying to identify that where their need is, and so yeah, I don't know if anyone else has anything to say about that as well. But yeah, and then I think
I might have a comment, Chris. Yeah, I was just going to say, I think another aspect that we encounter is the institutional support, the institutional context, the institutional environment. So if our space is deployed at a large university, there's huge variation in uptake, and a lot of that, most of it, I would say, really depends on how effectively the university is set up to act as an intermediary to introduce our space within its own environment. And I think it's quite fascinating. Some universities, the research data services people seem to have amazingly good relationships with their researchers, and the researchers trust them. I think that's kind of implicit in what Jens said as well. If the researchers don't trust you, they definitely won't work with you. And so in those cases, uptake can be amazing. In other cases, the universities either don't have the people, and that's really usually the problem. They don't have the resources, they don't have the data stewards, they don't have the structures. So they don't even have the communication channels to communicate with their researchers. So if there is a new tool, and we find in universities where our space has been deployed for 5 or 6 years, and people coming to us and say, "Oh, this is great, I never knew about it." Meanwhile, you talk to the research data services people, and they said they've been communicating it. Anyway, so I think the institutions as partners or not is also a critical factor in uptake.
thanks. And Anita, you have your hand up.
I kind of want to echo some of this. I mean, Jens hit the nail on the head when he said that there needs to be, your solution has to actually make the researcher's life easier, or you have to have just a very, very, very good reason, and then there's an uphill battle when using a PID. So we've tried really hard to hit at the journal level. And so, our PIDs are now available, or not available, but they're requested by about 1,000 different journals, which is, you would think that that's amazing, but that also has its drawbacks, because no one reads those kind of documents when they're submitting papers. So you really have to actually hit the researchers from multiple perspectives. And we've undertaken a big study, well, not very big study, okay, small study, trying to figure out when RIDs are successfully implemented in a particular subgroup versus others that are not. And we're trying to figure out, what are the best practices that actually help. And so with a lot of core facilities where we've been working with ABRF, which is the American Biomedical Resource Facilities, I think that's the right abbreviation, sorry. It's the number of core directors who are trying to advertise their services. And the core directors that are actually successful at getting people to cite them using the RID are actually doing multiple things. They have, they have their iLab system where their RIDs are at the top of the iLab system, requesting specifically that, their users cite them using the RID. They have it in their email communications as a line. They have it on their website. So And the more of these things that they do, it seems like the better compliance you get. So you actually really need to bring in a lot of partners, because any one channel is going to only hit a very small number of people. So I just wanted to kind of say that, yes, there is like maybe the most effective for some authors, but the most effective method to get to other authors is going to be different. So the more of those sort of channels that you actually hit with your persistent identifier, the better your overall compliance will be. So.
Yeah, and Anita, maybe I can follow what you just said. And in a previous life, I was a librarian. I still am a librarian, but I also now work on the society side, and you know, as the data steward, and I see what kind of impact now you know, some of our requirements have, our policies for you know, for instance, at the AGU, you cannot get published unless you share your data. And so then in those interactions, I see that, you know, authors sort of struggling with, what do I need to do to get my paper published? And in a similar sense, and it's a shame that Rowan's not here, but I think, you know, in some ways of what they're doing with the authoring process and looking at it ahead of time, even, you know, they in some ways authors can see, well, what do I have to do to meet the requirements of publication later on when I'm developing, you know, when I'm putting together my research paper. And so I think that driver is really compelling for researchers as well, is that, you know, they'll try to meet the requirements of the journals. And so I think that's why it's very important that we're all sort of aligned as well, that, you know, we're not just encouraging, you know, this, but actually, you know, with our guidance or our policies actually requiring that data is shared in software. And so I think like further on downstream, there's an opportunity, you know, for even driving, you know, these kind of activities, so the authors, you know, get better at doing this. So, but yeah, I don't know if anyone else has anything to add to that, but we might want to give people a little bit of time between the next session. I thought we were at the end of the day, but it continues on. I think this is a round the clock thing. Yeah, thanks everyone. Thanks to all of our speakers. It was really interesting hearing from all of you, and really appreciated hearing about your projects. And again, thanks to Rory for rounding us up and putting together this wonderful session. So thanks everyone. And we'll see you around at RDA and IDW around in some of the other sessions. So see you later. Take care.
Thank you.
Thank you.
Thanks,
Bye.
Chris.
Thank
Yep,
you.
bye-bye.
Bye.
Keep in contact.